12.2.2 Role of macro and micronutrients. Essential elements perform several functions. They participate in various metabolic processes in the plant cells, such as permeability of cell membrane, maintenance of osmotic concentration of cell sap, electron transport system, buffering action, enzymatic activity, and act as major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrient elements are given below. Nitrogen. This is the essential nutrient element required by plants in the greatest amount. It is absorbed mainly as NO3 negative, though some are also taken up as NO2 negative or NH4 positive. Nitrogen is required by all parts of plant, particularly the meristematic tissue and the metabolic active cell. Nitrogen is one of the major constituent of protein, nucleic acids, vitamins and hormones. Phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ion either as H2PO4 negative or HPO4 2 negative. Phosphorus is a constituent of cell membranes, certain proteins, all nucleic acids, and nucleotides, and is required for all phosphorylation reactions. Potassium. It is absorbed as potassium ion K plus in plants. This is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion cation balance in cells and is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in maintenance of the turgidity of cells. Calcium. Plant absorbs calcium from the soil in the form of calcium ions, Ca2 plus. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pactate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates in older leaves. It is involved in the normal functioning of the cell membranes. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by plants in the form of divalent Mg2 plus. It activates the enzymes of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is a constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate SO4 2 negative. Sulfur is present in two amino acids, cysteine and methionine, and is the main constituent of several coenzymes, vitamins, thiamine, biotin, coenzyme A, and ferredoxin. Iron. Plants obtain iron in the form of ferric ions Fe3 plus. It is required in larger amounts in comparison to other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferredoxin and cytochromes. It is reversible, oxidized from Fe2 plus to Fe3 plus during electron transfer. It activates catalase enzyme and is essential for the formation of chlorophyll. Manganese. It is absorbed in the form of manganese ion Mn2 plus. It activates many enzymes involved in photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as Zn2 plus ions. It activates various enzymes, especially carboxylases. It is also needed in the synthesis of oxygen. Copper. It is absorbed as cupric ions, Cu2 plus. It is essential for the overall metabolism in plants like iron. It is associated with certain enzymes involved in redox reactions and is reversibly oxidized from Cu plus to Cu2 plus. Boron. It is absorbed as BO3 3 negative or B4O7 2 negative. Boron is required for uptake and utilization of Cu2 plus. Membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plants obtain it in the form of molybdate ion MoO2 2 positive. It is component of several enzymes, including nitrogenase and nitrate reductase, both of which participate in nitrogen metabolism. Chlorine. It is absorbed in the form of chloride ion Cl negative along with Na positive and K plus. It helps in determining the solute concentration and the anion cation balance in cells. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution.